Hello, and welcome to the Clinical Care Options Infectious Disease Podcast. I'm your host, Jennifer Blanchette. Today's episode is part 3 of a 12-part series entitled COVID-19 Answering the Questions. This series features brief updates on the latest incidents and clinical data related to COVID-19 diagnosis and management, each followed by an in-depth question and answer session designed to address infectious disease specialists' most pressing COVID-19 questions. During this episode, Dr. Sharon Lewin from Melbourne, Australia, will provide an update on the latest COVID-19 incidence trends and a concise guide to management of mild and moderate disease. For more information on Dr. Lewin and for a link to additional online education from CCO's COVID-19 Comprehensive Resource Center, please visit the show notes for this episode. Now let's get started and hear a brief COVID-19 update and answers to clinician questions from Dr. Lewin.
Thanks very much, Jennifer, and great pleasure to be hosting and speaking at this webinar. As Jennifer mentioned, I'm based in Melbourne, Australia, where our epidemic is extremely small, and I suspect that most people on this call will have much more direct clinical experience of managing COVID-19 than I do, although as a virologist and an infectious diseases physician, I'm following this extremely closely. To put into context, Australia currently has just over 7,000 cases of COVID-19 with 100 deaths. So our outbreak has been very small and very different to many parts of the world, and particularly the US. If I could have the next slide, please, Jennifer. And this summarises where we are today. This was last updated on June the 22nd and comes from the Johns Hopkins database or global COVID-19 dashboard. And you can see here that currently the global cases are confirmed at just over 9 million. The largest number of cases still remains in the United States with just over 2 million. With Brazil rapidly increasing at over 1 million, and then around 500,000 in Russia and India, and then the UK. As I mentioned earlier, these numbers are very different to many parts of particularly the Asia-Pacific region, including Australia and New Zealand. The death rate is also approaching 500,000, which is extraordinary. Again, the highest hit countries include the US and Brazil, followed by the UK. These differences between confirmed cases and deaths will be a consequence of the pace of the disease, and we'll talk about that a little later, that most people, if they die, will die in the second or third weeks. So therefore, deaths will always lag behind infections and will also be dependent on the reporting structures in place in each country. And finally, the total numbers of cases recovered is also reported globally, sitting at about 4.5 million. And again, you can see that this number is significantly less than the number of confirmed cases because there is a lag with people taking at least 2 to 3 weeks, but if unwell, 4 to 6 weeks or longer to recover. Next slide, please. So here you can see a little bit of how the epidemic has unfolded showing this over time. And shown on the x-axis is the time between February 2020 and June 2020, where we are now, and the numbers of confirmed new cases in the thousands. And you can see here the 10 most affected countries with the 5-day moving average of daily confirmed new cases. And again, you can see the very early and rapid increase in the orange line representing the US, and a fluctuating number of new cases showing some decline towards early June, but then an increase from early June. Now, when you look at a diagram like this, of course, very helpful to know what's happening across the country, although many of you know, particularly those living in the US, that the outbreaks are very different in different parts of the country, with some cities and states reporting reducing numbers, some showing plateauing numbers and some showing increasing numbers. You can, though, get a sense of the tempo and uptick in cases. You can see here in the green line with Brazil, starting later than what we had seen in the United States. What looks like to be a slower increase, which can be affected by multiple factors, primarily, I suspect, the amount of testing being performed. And then you can see this slow and steady increase with no evidence of plateau. The other countries in here are, of course, a little harder to see because of the scale and because of the different tempo of those outbreaks. But I think this shows you that we have considerable challenges in both North and South America currently. Next slide, please. So I'm going to now switch to management of mild to moderate COVID-19. Next slide, please. So summarised here are definitions that come from the WHO on the left and the National Institutes of Health on the right for the medical management of mild COVID-19. And the medical management of mild COVID-19, which usually means that the person is not hospitalised and not requiring any oxygenation, or their oxygen levels are greater than 94 millimetres mercury. Hospital interventions are typically unwarranted, but isolation required to contain SARS-CoV-2 transmission. I should add there that in some countries, people with mild COVID-19 are indeed admitted, but this is largely to contain transmission and isn't required medically. And in the context of a large outbreak, is definitely not advised given the access to limiting resources. The WHO also recommends just treatment of symptoms, which includes antipyretics for fever, educating people on the signs and symptoms of complicated disease, so that if they deteriorate, they should promptly pursue urgent care. And this is actually very important because people can deteriorate and have advancing severe illness over a very short period of time. You can see on the right are the recommendations from the National Institutes of Health, saying that the majority of cases should be managed in an ambulatory setting or at home, and they may have some contact through telemedicine, which is shown to be helpful. Close monitoring advised as rapid progression possible, and the way to deal with this is by education, and that no specific laboratory tests are indicated if otherwise healthy. Finally, the NIH recommend that anticoagulants and antiplatelet therapy should not be initiated in non-hospitalised patients to prevent deep venous thrombosis or arterial thrombosis unless there are other indications. Next slide, please. This slide summarises the medical management of moderate COVID-19. In the setting of moderate COVID-19, the recommendation is to admit to a healthcare facility for observation, to administer empiric antibiotics if bacterial pneumonia or sepsis is strongly suspected, and that this should be re-evaluated daily, and as in all practice, to de-escalate or stop treatment if no evidence of infection. A critical component of the management of moderate COVID-19 in hospitals is the infection control management, and that includes limiting the numbers of individuals and providers entering patient room. And that will vary in different jurisdictions and different hospitals of how that is set up. But most hospitals will have dedicated COVID-19 teams and limiting entry into the patient room. To use protective measures for aerosol generating procedure, and they should include staff wearing an N95 respirator or a personal respirator compared to surgical masks. Surgical masks are warranted for other non-standard care or non-aerosolized generating procedure. And the WHO recommends that isolation location in either a health or community facility or at home really depends on the clinical presentation and how much supportive care is required, and that hospitalisation is preferred for those at high risk of deterioration. On the initial evaluation, this will, in someone with moderate COVID-19, who most likely will be admitted to a healthcare facility, one would request a chest X-ray, potentially ultrasound or CT of the chest, an ECG if indicated, a complete blood count with differential and metabolic profile, including liver and renal function, and one may choose to also measure inflammatory markers such as the CRP, D-dimer or ferritin. These are unlikely to change clinical practice, but may be valuable for predicting prognosis. Next slide, please. This shows, I think I like this figure quite a lot because it gives you a sense of the overall natural history of COVID-19. It's a little bit complicated, but I'll take you through this. It shows you the tempo of the illness and how symptoms are sequenced related to severity. On the x-axis, you can see the days since COVID-19 symptom onset. And on the y-axis, and shown in the orange line, is an estimate of the viral load of SARS-CoV-2 RNA in throat swabs. And this is a semi-quantitative assay. So if you just look at the orange line and look over the natural history of someone with prolonged illness, so over the first 3 to 4 weeks of days since symptom onset, you can see that viral load actually peaks early in the illness. And in fact, many data now suggest that viral load is maximal 1 to 2 days before the onset of symptoms. So you can see that orange line heading up and then slowly heading down or reducing viral load over the course of the illness. So there is a disconnect with how much virus there is and the severity of the illness. High viral load doesn't necessarily mean more severe disease, as we might think of in other conditions, specifically HIV. So in the first week of days since symptom onset, symptoms are often in the more mild range: fever, cough, myalgia, dyspnea. You may judge this clinically as mild disease. And there seems to be progression in symptoms and deterioration, commonly in the second and third weeks, which is when hypoxia, respiratory failure, hypotension, or persistent fever might be observed. And you can see that in the mid-range blue box on the x-axis between days 8 and days 14. And then if people are going to not improve, and of course at any point in time people can improve and symptoms resolve, but if there's disease progression, it's in the third week that the individual may develop ARDS or acute respiratory distress syndrome and either die or recover. And understanding this tempo or natural history of the infection is really important when thinking about interventions. Again, quite different to HIV. Viral load is not the entire cause of the problem here. People tend to get sick in that second and third week due to immune dysregulation, and we're now learning a lot more about what components of the immune system trigger this clinical deterioration and disease severity. But most work points to this being driven by a cytokine storm, particularly mediated by IL-6 or interleukin-6 and the innate immune response. Through understanding this disease pathogenesis, it's given us a clearer rationale for treatment choices. And as we are seeing more and more treatment results of clinical trials being published or announced, we're getting a much better understanding of which treatment should be used at what time. Again, this is very different, I think, to our understanding, and I use HIV as an example primarily because it is relevant, but also it is my own area of expertise, where antivirals for HIV play a very important role at any stage of the disease. You control the virus, and then the disease is under control and CD4 T cells recover. In COVID-19, this is quite different. The virus is causing, we think, most of the issues early on in the natural history, while a dysregulated immune response is responsible for clinical deterioration and more severe respiratory disease. So underneath the time course, you can see that a schematic, and this is just conceptual and it's not absolute, of when particular interventions are best used. And by the type of triangles you can see that interventions that target the virus are best used early in the natural history, with the goal of stopping the patient deteriorating into the second and third phases. So you can see here antivirals or small molecules that we'll talk more about are important. Convalescent plasma, which is collecting plasma from someone that's fully recovered from COVID-19, has high levels of neutralizing antibody, screened for any other infectious disease, and then given to a recipient, of course, hasn't yet been demonstrated in randomized clinical trials to show benefit, but high interest in convalescent plasma essentially acts as an antiviral drug because you're giving someone else's antibodies. And you can see that immunomodulation, or strategies to reduce the dysfunctional immune system, reduce cytokines such as IL-6, really play a role in this middle phase when people have more moderate or severe disease. Now, how antivirals should be combined with immunomodulators, when immunomodulators will have their maximal effect, we're only just beginning to learn about that. And finally, adjuvants, which would include symptom treatment or treatment for hypercoagulation, could potentially be used at any stage of the illness. And so the optimal timing is still a very important aspect for us to fully understand. And I think this slide is important because it also tells you when you interpret clinical trials, you need to think carefully about the inclusion criteria and the clinical profile of participants and which stage they are at in this natural history of COVID-19. Because interventions that may work for someone with severe disease or ARDS will likely be and have already shown to be quite different to what might be of benefit in someone early on in the disease or with mild disease. Next slide, please. So here are the NIH guidelines of what and how to use COVID-19 treatments, which are currently considered investigational. And I recognise that there are non-US people on this webinar, including myself, and that each country will have very different COVID-19 treatment guidelines. So I'll just acknowledge this up front, but we'll focus on the NIH guidelines. So as the previous slide showed, we think about these interventions as targeting the virus, antiviral treatments, or targeting the immune system, or immunomodulators, or immune-based therapies. And eventually we may learn that these could and be used together, though those data are not yet available. So in the NIH guidelines, remdesivir, and I'll talk to you a little bit more about remdesivir, a direct-acting antiviral, which works by inhibiting RNA-dependent RNA polymerase, is recommended for hospitalised patients with severe COVID-19. Then NIH says there's insufficient data to recommend for or against patients with mild to moderate COVID-19. And there are now many studies currently underway, should be complete quite soon, addressing this question, and I'll talk about those later in the talk. The NIH actually recommends against high-dose chloroquine. That means 600 milligrams twice a day for 10 days. And this is because there has been no evidence of any activity of high-dose chloroquine, but there has been evidence of toxicity and increased risk of adverse events, so recommended against using high-dose chloroquine. There are a number of experimental antiviral agents that the NIH recommends against, except in the context of clinical trials. And many of these agents you would have read vastly conflicting ideas about whether they work or not. And there's emerging data that should be available very soon in large randomized controlled trials that include a spectrum of people with disease severity to actually answer these questions. Those clinical trials will be available, results of those clinical trials in peer-reviewed publications should be available soon. But at the moment, the recommendation is that this should only be used in the context of clinical trials. And these include hydroxychloroquine, highly politicized drug, many studies underway, early announcement of a large randomized controlled trial a few weeks ago showing no benefit of hydroxychloroquine from the recovery group in the UK, but those data still not available in publication. Hydroxychloroquine and azithromycin is also recommended against, except in the context of a clinical trial. Lopinavir, ritonavir, or Kaletra, or any other HIV protease inhibitors are being actively investigated, still in clinical trials. The recommendation is to not use these outside of clinical trial. Now, immune-based therapies are really still emerging of how best to use them. Some exciting data on dexamethasone, which would fit into this bucket, which was announced by the recovery trial in the UK about a week ago, and in fact their publications in bioarchives in the last 12 hours, overnight for me. So unfortunately I haven't been able to scrutinise it, but I will talk to you a bit about dexamethasone. And so the guidance is that there's insufficient data to recommend for or against a number of immune-based therapies. They include convalescent plasma, which I think of as actually an antiviral agent because it contains antibodies targeted against the virus. SARS-CoV-2 immune globulin, which means purified immune globulin from someone that's recovered from SARS-CoV-2. IL-1 inhibitors or IL-6 inhibitors, such as tocilizumab. And there again are currently clinical trials assessing each of these interventions, and there currently is insufficient data to recommend for or against. The NIH recommends against the use of a number of agents except in the context of clinical trials, and that includes non-SARS-CoV-2 specific intravenous immunoglobulin or IVIG, or immunomodulators such as interferons or JAK-STAT inhibitors, which are also now being evaluated in clinical trials. I should just add that severe in the definition here used by the NIH and widely used in many clinical trials is defined as a blood oxygen of less than 94% on ambient air at sea level, and therefore requiring supplemental oxygen, mechanical ventilation, or ECMO. And keep that in mind, particularly when reviewing the results of clinical trial data. And in the last few slides, I'll give a brief update of some further investigational agents. Next slide, please. So this is a really important study. This is the randomized evaluation of COVID-19 therapy or the recovery trial being run in the UK amongst hospitalised patients. In this study, which has enrolled many thousands of people in an extraordinarily rapid time, patients are randomised to standard of care with no additional treatment, with lopinavir, ritonavir, or Kaletra, dexamethasone, hydroxychloroquine, or azithromycin, so a 5-arm study. They also have a factorial design with simultaneous allocation to no additional treatment versus convalescent plasma, And if progressive disease or a hyperinflammatory state, subsequent randomization to no additional treatment versus tocilizumab on anti-IL-6. The study has enrolled more than 11,000 patients from over 175 hospitals in the UK, and they enrolled this in actual record time. I saw recently an interview with the lead investigators saying that the study was designed and approved within a week, which is quite extraordinary. And they've made, the recovery trial have decided to announce their results ahead of publication through press releases. This has been a pattern we've seen over the last few weeks or months. It's understandable in the middle of an epidemic, but of course, I think one always, at the middle of a pandemic, I think one always needs to be a bit cautious in just looking at the press announcement, because the complexity of the participants is not always apparent. So on June the 5th, the recovery group announced that they were closing recruitment to the hydroxychloroquine arm because of lack of clinical benefit. They reported the 28-day mortality was 25%, which I should say is high, but these are all hospitalised patients, of course, so this mortality rate is high. With hydroxychloroquine and standard of care, and there were 1,500 people that received that intervention versus 23.5% with standard of care alone, and over 3,000 received that intervention, with a relative risk of 1.11, with confidence intervals crossing 1, making this not significantly different. And these results have been announced, the publication not yet available. And many studies around the world that are using hydroxychloroquine, and there are many others. And I should point out that this is the largest randomized study to date of hydroxychloroquine, the recovery trial. There are many hundreds of studies of hydroxychloroquine that actually are still enrolling and are waiting with great interest to see these results published. Their second announcement, which was very exciting, was on June the 8th, where they announced that recruitment to the dexamethasone arm had halted because sufficient patient numbers had been enrolled to establish potential and benefit. And I think many of us were most excited to see this result. The next slide, please. And here are the details are reported by the press release. The paper is now available as on bioarchives as a pre-publication, so still not peer-reviewed, but all details are now available. And this was a very interesting finding, and I think unexpected by many people in the field. First of all, the dose of dexamethasone used here was 6 milligrams a day for 10 days, oral or IV, and compared to standard of care, which just had no additional steroids. And they assessed their findings, and again, the primary endpoints also very important in determining success. Many studies to date have just assessed clinical improvement or reported statistical benefit in clinical improvement. This is the first study to show a significant effect on 28-day mortality, a far harder endpoint. And they divided their analyses into people requiring, just starting from the bottom, no respiratory intervention. So they were assumed to have mild to moderate disease but hospitalised. Patients requiring oxygen only, intranasal oxygen only, and then patients requiring ventilation. And you can see here, and the numbers are now, you'll be able to find in the study, in the actual preprint, these numbers are large. There were 2,000 people that received dexamethasone and 4,000 standard of care alone. And then the third column, the relative risk of death for dexamethasone and standard of care versus standard of care alone with 95% confidence intervals. So you can see that for patients requiring ventilation and patients requiring oxygen only, so this is severe disease, there was a significant benefit with a 35% reduction in death in people requiring ventilation with the addition of dexamethasone, and a 20% reduction in death in people requiring oxygen alone. So these are pretty spectacular results. And as I said, the first randomized clinical trial to show an effect on mortality, which was highly significant. And interestingly, no effect of dexamethasone in patients requiring no respiratory intervention, no reduction in the relative risk and the confidence intervals crossing 1. Next slide, please. And that brings me to the end of the formal presentation, and happy to take any questions.
Great. Okay, sorry about that. Yes, great. Thank you so much, Dr. Lewin. So we have had quite a few questions, and we'll try to get through as many as we can now. Susan asks, after obtaining a positive nucleic acid amplification test result, is antibody testing needed as a follow-up in the weeks afterwards?
In Australia, and I think similar to other countries, we would not recommend antibody testing as follow-up. Antibody testing, and we did a clinical care options a presentation just recently, although this field is changing quickly at the moment, is not recommended for diagnosis. No need. Once you have a positive PCR test, you have COVID-19.
Okay, great. Foster asks, a new study was published in Nature showing that immunity is transient, but it was a small number of patients. Are there more studies looking at that? And also, what are your thoughts on that finding?
First of all, it's important how you define immunity. At the moment, we don't know whether having COVID-19 once protects you from reinfection. Most likely it does. The immune response to COVID-19 includes generating antibodies, which are detected in nearly everyone by about day 14 and certainly by day 21. You can detect IgG or IgM antibodies. The key factor that we think is important is the development of neutralizing antibodies, meaning antibodies that actually neutralize and eliminate the virus. And you can quantify those quite accurately too. And there's now several studies showing that nearly everyone develops neutralizing antibodies, although their titer will vary. And there's a direct relationship between total antibodies and neutralizing antibodies. What we don't actually know is what level of neutralizing antibodies is critical to prevent from reinfection. And I think we will, or protect from infection de novo, very important in vaccine design. So I think we're going to learn that with bigger studies that look at serology, bigger studies that look at natural history, animal studies. The idea that immunity wanes, I'm actually not familiar with that study. The work that I have seen shows that your antibody titer increases progressively and maximizes at day 21. And I haven't yet, and similar neutralizing antibodies, I haven't seen evidence that that actually declines. From other work on other coronaviruses, especially in animal models, it's not a given that immunity is lifelong, as you might have following measles, for example. But to date, I haven't seen any evidence of when titers of neutralizing antibodies decline and how often that happens.
Okay, great. Jamila asks, since dexamethasone showed a benefit, do you think other steroids like methylprednisolone or hydrocortisone might also work?
Yes, a good question. I suspect so, although most of us prefer to focus on evidence-based recommendations. At the moment, the study that has shown these dramatic effects are with dexamethasone. So my recommendation would be to stay with dexamethasone, but as you know, we do use these interchangeably, for example, with how we might manage severe pneumocystis pneumonia. I'm sure there'll be other studies that will come out that will tell us whether prednisolone gives the same effect. My suspicion is probably not. My preference is follow the evidence and use the dose and duration that was shown to work. And I'm sure we'll refine that over time.
Okay, great. A question from Viral. Where does dexamethasone fit into the treatment algorithm? Will it be administered to all individuals requiring oxygen ventilation? So kind of the point. Yeah.
Yeah, I think everyone's now will be amending and updating their guidelines fairly rapidly as soon as the peer-reviewed publication is available on dexamethasone. I think it fits in the treatment guidelines, and I say this with some reservation because, of course, the publication is not yet peer-reviewed, and I would like to see that first. But based on the data announced and in the preprint, the benefit is only in people with severe disease requiring either oxygen or ventilation, and that's where you see the benefit, not in people with mild disease. Again, this is a little bit reminiscent to me of PCP, pneumocystis carinii pneumonia, where steroids played a major role only with people with severe disease.
Okay.
Now, how these interventions might be used in combination with antiviral drugs, for example, other immunomodulators, those we'll get insight into that, I'm sure, with future studies.
Okay, great. We've had a couple of questions on your thoughts on other potential therapeutics, umifenavir and ivermectin. Do you have any thoughts, or have you seen any data on the use of those agents?
I'll start with ivermectin because it comes from our institute.
Okay.
And it's a discovery from Melbourne, actually led by colleagues at Monash University, another major medical school in Melbourne where I went to medical school. And investigators at Monash University have been interested in ivermectin we all know ivermectin is an anti-parasitic drug, but ivermectin actually also has antiviral properties. And the group at Monash University have been studying this for many years, looking at its effect on dengue and HIV, largely related to nuclear import. Ivermectin seems to block entry of viruses like dengue and HIV into the nucleus. So, or passaging through the cytoplasm. And so that when COVID-19 was discovered, they rapidly asked the question, does ivermectin also block entry of COVID-19? COVID-19 doesn't need to enter the nucleus for its replication. And they partnered with investigators from my institute, for the Doherty Institute, as we have the PC3 capabilities to grow the virus and look at antiviral efficacy, and show that ivermectin did have antiviral properties. And this was published the only work that's been published is in vitro. And unfortunately, the dose used in these studies in vitro was much higher than you can achieve with the current dosing of ivermectin. And so although it was interesting, exciting that there was a drug that had antiviral activity in vitro, not yet shown in animal models, I should add. It was at a very high dose, not feasible to achieve with current dosing strategies for ivermectin. So I definitely think ivermectin needs a lot more work. They're still working away at this, about whether its activity at lower doses, methods of delivery, similar drugs to ivermectin, whether they may have greater potency. So I'm not optimistic that we will find that ivermectin has potent antiviral activity, but it's further work's needed. I should just add, though, that there was a large study that came out quite soon after that in vitro work, which was published, I think, in Antimicrobial Chemotherapy, showing that ivermectin had a survival benefit. But I would highlight to you that that publication was only in preprint and actually came from a database that there is some question about, the SurgiSphere database, which subsequently led to the retraction of articles on hydroxychloroquine from Lancet and ACE2 receptor blockers from the New England Journal of Medicine. So we don't even know if the SurgiSphere database is real or not. So I would ignore that clinical paper on showing there was a survival benefit of ivermectin. I would be cautious about its role in. I would definitely not be using it in any form outside a clinical trial, and it's not even in clinical trials yet because the dosing still needs to be worked out. And the other one was umifenavir, which is a anti-influenza drug. So as you will all know, influenza is an RNA virus and shares some pathways common to SARS-CoV-2, although they're obviously very different family and different replication cycles. And uviprenavir, if I've got that right, is an anti-flu drug developed and I think licensed for influenza in Russia. Not available outside of Russia. And there has been some small studies looking at uviprenavir in China. So when COVID-19 hit China, there was a flood of small clinical trials looking for things that worked. Understandable. Pandemic, totally new virus, pretty severe consequences, and people were looking for proof of concept. And so there were a lot of small studies that came out that showed potential proof of concept that this might work. China actually adopted some of those interventions into their clinical guidelines. In the early days, again, I guess that's sort of acceptable and not sort of, it's acceptable. You're in a pandemic and you don't know what is working. But we're in a different phase now. We're now 5 months, 6 months into the pandemic with a lot of clinical data now coming out. And so I think we need large randomized controlled studies to know what works and what doesn't work. And that has not been done with uviprenavir. And actually, I'm unaware of a large RCT. I bet you it's out there, but I'm unaware of, it's not included in these big platform trials running out of the UK, US, and we have one such study in Australia as well. So I'll just say one last thing about the importance of RCTs. I'm sure I don't need to convince this audience of infectious disease physicians of the importance of evidence base, but the kind of preclinical hunch early in COVID-19 or clinical suspicion was that dexamethasone would not play a role. And that came from experience with SARS-1, I should add, not from controlled clinical trials, from observational studies, that dexamethasone was thought to make people worse, but it was never examined in an RCT. And so here you now have a proper RCT, very large numbers of people randomized with a clear mortality benefit. And so I think this is a lesson, though we all know it, but it's a reminder that you need the large RCT to answer the question properly. And we may, or looks like we now have at least one intervention that will dramatically reduce mortality. And we only got this because of an RCT. The clinical suspicion, as I said, or it was that dexamethasone or steroids was not going to work for coronaviruses. So yes, these experimental agents are really important. We need to test them in studies. We need to test them alone and in combination in an appropriate size to work out if they work or not.
Great. Okay. I think we have time for one last question, and this pertains to, someone has asked about the second outbreak. Is it likely to happen? And it ties to another question, which is really what differed in Australia that from other places like the US or Italy that has allowed the disease to be, or the virus to be, much better controlled?
The key difference was detection and testing early in the outbreak. I've absolutely no doubt that that's been a major, major factor. So in Australia, we developed a test for coronavirus at our institute, the Doherty Institute, led by Dr. Mike Catton, in around mid-January. As soon as the reports of this new coronavirus were made out of China, we became aware of it, of course, in early January, that sequence that it was a coronavirus published in January the 10th. And by mid-January, we had a test for this coronavirus that we then began to share with other similar laboratories around the country. Of course, everyone's public health system differs. In Australia, we don't have a CDC, but we have what's called public health reference laboratories in each state. So sort of 6 major public health reference laboratories. So we were testing people. We had a test available by late January. Our first case was diagnosed in Melbourne at the Doherty Institute by Mike Catton on January the 24th, 2 days after the first diagnosis was made in the US on January the 22nd. By that time, on January the 24th, we, as I said, had a test up and running. we were testing every person coming from China. But we're not testing everyone. We were heightened awareness of clinical illness in people coming back from China. We stopped flights from China to Australia on February the 1st, actually, I think about 24 hours later than the US. But the difference was that we were testing anyone with clinical symptoms who had an epidemiological risk factor at the time, coming from China or a close contact with the case. And those epidemiological risk factors, of course, changed over time as we saw an outbreak in Iran and northern Italy, and then, of course, the US. So the key for us was that we were testing, isolating infected people and quarantining all their contacts very aggressively from about mid to late January. Mm-hmm. And if I actually going back to those numbers, our testing rates in February were extremely high. And I think countries that have done well have had those testing rates, effective public health measures of isolation and quarantine, and as well as travel bans in place and operational very early. And a week can make a huge difference. I'll just say one last thing. People often say to me, oh, Australia's an island, it was summer, you were protected. But it actually was not the case. By mid-March, our numbers were doubling every 3 to 4 days, just like they were everywhere else in the world. Mm-hmm. They peaked in March the 29th. By that stage, we already had a lot of testing, so we were diagnosing a lot more people and therefore aware of who was infected and isolating quarantine. March 29th, we went into lockdown, and so then the physical distancing, of course, has played a major role. And although we have really small numbers, we still have a lot of physical distancing here in Australia. So for me, for example, in my work, we've only been working at 30% capacity. Restaurants only opened about 10 days ago. So in summary, early and aggressive testing, high awareness that this was a problem, travel bans, and then physical distancing. And the compliance has been pretty good. And starting early was the difference.
Thank you very much, Dr. Lewin, for that helpful information. And thanks also to our listeners. As a reminder, to view the COVID-19 Comprehensive Resource Center on the Clinical Care Options website, click on the link in the show notes. Also, please be sure to check back regularly for more episodes on important infectious disease topics. Thanks again for listening.